Assalamu alaikum class. My name is Ramsul, roll number 5017. My topic is tumor viruses. As the name suggests, as the tumor viruses, those viruses which cause the cancer in the various organisms. It includes the six distinctive families of the animal viruses called the tumor viruses. Out of five families are the DNA genome viruses, which are referred as the DNA tumor viruses, and sixth family of these tumor viruses are the retroviruses, which are basically RNA genome virus particles. These are replicate via synthesis of a DNA provirus in infected cells. Now we will discuss DNA tumor viruses. Here are the virus families and their causing tumor, and also genome size is written in the table. And now we will discuss about the hepatitis B virus, HBV virus, which is the smallest genome of 3 kb of animal DNA viruses. It infects liver cells, species in the various species including ducks, woodchucks, squirrels, and also including humans. Hepatitis B virus infection is particularly common in the parts of Asia and Africa where it is associated with up to million of cases of the liver cancer annually. In addition, development of cancer induced by the HPV virus is driven by a continual proliferation of liver cells that results from the chronic tissue damage. Then we will discuss about the SV40 and polyomavirus. These virus is associated with the human cancer. They have been critically important as models of understanding the molecular basis of the cell transformation. SVV40 and polyomavirus do not induce tumors or transport cells of their natural host species, monkeys and mice respectively, in cells of their natural host, which is primitive cells. Infection leads to the virus replication, cell lysis, and release of the progeny of the virus particles. Both SV40 and polyomavirus early region proteins induce transformation by the interacting with the host proteins that regulate the cell proliferation. And next is papillomavirus. The papillomavirus are the small DNA viruses which have approximately 8 kb genome that induce both benign and malignant tumors in humans and a variety of other animal species. Cell transformation by the human papillomavirus results from the expression of two early region genes E6 and E7. E6 and E7 proteins act analogously to SV40 T antigen by interfering with the function of the cellular RB and P53 proteins. In particular, E7 binds to the RB and E6 stimulates the degradation of the P53 by ubiquitin mediated proteolysis. Next is the adenoviruses. The adenoviruses are large family of DNA viruses with genome of about 35 kb. In contrast to the papillomaviruses, the adenoviruses are not associated with the naturally occurring cancers in either humans or other animals. Transformation by the adenoviruses results from the expression of two early genes, E1A and E1B, which are required for virus replication in primitive cells. Then next is herpes viruses. Herpes viruses has genome size of 100 to 200 kb range. It induces tumors in animal species including frogs, chickens, and monkeys. In addition, two members of herpesviruses family, Kaposi's sarcoma-associated herpes viruses and Epstein-Barr viruses, are associated with the human cancers. Next is RNA tumor viruses, which are basically also known as retroviruses. These viruses have RNA as a genome. One of the retroviruses is human T cell lymphotropic virus type 1, HTLV1, is the causative agent of adult T cell leukemia, which is common in the parts of Japan, the Caribbean, and Africa. Transformation of T lymphocytes by the HTLV1 results from expression of the viral gene, which encodes a regulatory protein affecting expression of the several cellular growth control genes. AIDS is also caused by another retrovirus which is known as human immunodeficiency virus.